Good morning, students. Today, let us discuss about the chapter Life Processes in Biology. Now, how can we tell that what is alive and what is not alive? If you see a dog running, or a cow chewing a cud, or a man walking on the street, we know that these are living beings. But what about plants? How do we know that they are alive? We see them green, and we see them grow over time. So we know that they are alive. Now, the criteria to decide whether something is alive. The most important criteria to decide whether something alive is movement. All living things move with the help of any external help. Some movements are easily visible, like movements of body parts, like walking, running, lifting our hand, like whatever the work we do, we can see the movement. Some movements are not easily visible, like molecular movements. Movement of molecules we cannot see with our naked eye. The molecular movements in the cells and tissues is necessary for all life processes. Now, let us discuss about life processes. Life processes are the basic processes in living organisms which are necessary for maintaining their life. The basic life processes are nutrition, respiration, transportation, and excretion. Nutrition is the process of taking food by an organism and its utilization by the body for life processes. Respiration is the process of taking food by an organism. Respiration is the process by which food is burned in the cells of the body with the help of oxygen to release energy. Whereas transportation is a process by which food, oxygen, water, waste products are carried from one part of the body to the other. Excretion is a process by which waste products are removed from the body. Now, about nutrition. Nutrition is the process of taking food by an organism and its utilization by the body to build the body, for growth, to repair the damaged parts of the body, and for energy. Life on Earth depends on carbon-based molecules, and most of the food are also carbon-based molecules. The outside raw materials used by the living organisms are food, water, and air. Modes of nutrition. There are two main modes of nutrition. They are autotrophic nutrition and heterotrophic nutrition. Autotrophic nutrition. It is a nutrition in which organisms prepare their own food from simple inorganic substances like carbon dioxide and water in the presence of sunlight and chlorophyll. Example, all green plants and some bacteria. Heterotrophic nutrition. It is a nutrition in which organisms get their food directly or indirectly from plants, all animals, fungi, and some bacteria. Types of heterotrophic nutrition. There are three main types of heterotrophic nutrition. They are saprophytic, parasitic, and holozoic nutrition. Now about saprophytic nutrition. It is a nutrition in which organisms get their food from dead and decaying organisms. They break down the food material outside their body and then absorb it. Examples, mushroom, bread mold, yeast, some bacteria, etc. In the picture you can see the mushrooms. They take the food from the dead and decaying organic substances and they grow. Now about parasitic nutrition. Parasitic nutrition is the nutrition in which organisms get their food from living organisms without killing them. Example, cuscuta. So you can see in the picture cuscuta. So see, it is a thread-like structure around the plant. So the plant which provides food to the cuscuta is known as host plant. It does not affect the host, but it takes food from the plant. So examples for parasitic nutrition is Cuscuta, orchids, etc. Now about holozoic nutrition. Holozoic nutrition is a nutrition in which organisms take food directly and then digest and absorb it. Example, amoeba, paramecium, birds, fishes, human beings, etc. Now about nutrition in plants. How the photosynthesis takes place, let us discuss. Photosynthesis is the process by which plants prepare food by using carbon dioxide and water in the presence of sunlight and chlorophyll. The food prepared is carbohydrate, which is stored in the form of starch. Oxygen is released in this process. Now, equation for photosynthesis: 6CO2 plus 12H2O gives rise to C6H12O6 plus 6H2O plus 6O2. Here, the plants they will utilize carbon dioxide and water and produce glucose, and oxygen is liberated. So, in the presence of sunlight and chlorophyll. So, process of photosynthesis generally takes place in three main steps. They are absorption of light energy by chlorophyll, conversion of light energy into chemical energy, and splitting up of water molecules into hydrogen and oxygen. Reduction of carbon dioxide by hydrogen to form carbohydrates. In this picture, you can see the picture here. The plant takes carbon dioxide and water and prepares food. The food is in the form of starch. So generally the chlorophyll traps the sunlight. Here the light energy is converted into chemical energy. Now let us know in detail about chlorophyll. Chlorophyll is nothing but a green pigment present in the leaves. If you observe a cross section of a leaf under a microscope, we can see the cells containing green dot-like structures. These are called as chloroplasts, and these chloroplasts contain chlorophyll. Now, we'll closely observe the transverse section of the leaf. So the transverse section of the leaf contains upper epidermis, mesophyll, lower epidermis. Upper epidermis is coated with a waxy cuticle outside, and in between the upper epidermis and lower epidermis, mesophyll tissue is present. It is of two types: palisade mesophyll tissue and spongy mesophyll tissue. Palisade mesophyll tissue, the cells are closely arranged. In the spongy mesophyll tissue, 8 spaces are present. The green colored dots you can see in the mesophyll tissue, they are nothing but the chloroplasts. Now here you can see the plant cell. In the previous classes, we have discussed about the plant cells. So plant cell consists of a cell wall made up of cellulose, and under that cell membrane is present. Most of the plant cell is occupied by a vacuole. So vacuole is bigger when compared to the animal cell. And the nucleus, cytoplasm is present inside the cell membrane. Now you can see a chloroplast in a circle. So that is a chloroplast which contains a chlorophyll. Now, if you see the chloroplast under electron microscope, it looks like this. A chloroplast contains two membranes, outer membrane and inner membrane. In between the two membranes, a membranous space is present, which is called as intermembrane space. Inside the inner membrane, a aqueous fluid-like substance is present, which is called as stoma. And you can see the circular one. So in that, a stalk of thylakoids are present, which are called as grana. Now, here, stomata open to allow carbon dioxide to enter a leaf and water vapor to leave. Generally, stomata performs two functions, like in taking of carbon dioxide and in the removal of excess of water. Now let us know in detail about stomata. Stomata are the tiny pores present in the leaves through which exchange of gases takes place. Each stomata has a pair of guard cells which controls the opening and closing of stomatal pore. When water enters the guard cell, it swells and the pore opens. When the guard cells lose water, it shrinks and pore closes. Now here you can see the stomata under electron microscope. It looks like this. Now see here, water diffuses into the guard cell which causes them to open. On hot dry day, the guard cell have less water, they relax and stomata closes. Now let us perform an activity to show that chlorophyll is necessary for photosynthesis. Take a potted plant with variegated leaves. Variegated leaves means the leaves which has patches over it like white and green, yellow and green. So for example, if you take a money plant or a croton, you can see the variegated leaves. Now keep the plant in dark room for 3 days so that all the starch get used up. Now keep the plant in sunlight for about 6 hours. Pluck a leaf from the plant. Mark the green areas in it and trace them on a sheet of paper. Dip the leaf in boiling water for few minutes. After this, immerse it in a beaker containing alcohol. Now carefully place the above beaker in a water bath and heat till the alcohol begins to boil. What happens to the color of the leaf? What is the color of the solution? Now dip the leaf in a dilute solution of iodine for few minutes. Take out the leaf and rinse off the iodine solution. Observe the color of the leaf and compare this with the tracing of the leaf done in the beginning. So what can you conclude about the presence of starch in various areas of the leaf? It will be seen that only the green parts of the leaf turns blue-black. This shows that chlorophyll is necessary for photosynthesis. Now, let us perform another activity to show that carbon dioxide is necessary for photosynthesis. Take two healthy potted plants which are nearly the same size. Keep them in a dark room for 3 days. Now place each plant on separate glass plates. Place a watch glass containing potassium hydroxide (KOH) by the side of one of the plants. The potassium hydroxide is used to absorb carbon dioxide. Cover both the plants with the separate bell jars. Use Vaseline to seal the bottom of the jars to the glass plates so that the setup is airtight. Keep the plants in sunlight for about 2 hours. Pluck a leaf from each plant and check for the presence of starch as in the above activity. Now, do both the leaves show the presence of same amount of starch? What can you conclude from this activity? The leaf of the plant kept in the jar containing potassium hydroxide does not show the presence of starch. This shows that carbon dioxide is necessary for photosynthesis. Students, in the next class, we will discuss about heterotrophic nutrition. Thank you.